Good morning, everyone. I welcome you all to the morning session of the day. And today we have the honor and the pleasure of having Dr. J.K. Sundray with us. Sir is H.O.D. Fish Genetics and Biotechnology at ICAR-Central Institute of Fishery Water, Central Institute of Freshwater and Aquaculture, Bhubaneswar, Odisha. And the topic chosen by our esteemed guest for deliberation today is aquaculture, genetics, and genomics. Sir, you are audible as well as visible, and your screen is also visible. You may start, sir.
Yeah, good morning, all of you, wherever you are there in virtual mode. Thank you, the organizer, to giving me chance to talk about aquaculture genetics and genomics. My outline of presentation will be like, I'll be talking a little bit about my institute and what is the aquaculture status in India, hunger and food security, his genetics, his genomics, and action need to increase the sustainable aquaculture production. It is more of a generalized presentation, but if you have any specific questions, you can come back to me. All of you might be knowing about what is United Nations 17 goals for sustainable development. If you look into fisheries and aquaculture, we have been playing a major role in many of these SDGs, but our major focus has been no poverty, zero hunger, life below water, and the climate action. Right? So, how fisheries as a product or byproduct, aquaculture as a product or technology is helping to reduce the poverty and reduce the hunger. And we have been talking about this, the freshwater, brackish water, marine water, life below water has been one of the prime SDG for the aquaculture fisheries people. And the research about the climate action, you might be knowing about the climate action, how the climate has been playing a major role in the, poor performance of the aquaculture. And apart from this, sometimes we also work into quality education. Maybe we are not directly related as a fisheries research institute, but many of our faculty, they do a lot of training program. So this is number 4 SDG also we are targeting. ICAR-CIPA, you might be knowing, this is based in Bhubaneswar, and we are one of the regional lead center for NACA. And the focus mostly we are working in the basic strategic research, diversification, and human resource development. And if you look at the vision and mission, we have a technology or we are to develop technology which is global competitive. Okay. And why genetics and genomics has been important in the aquaculture or in the food security. If you look into these two reviews, one editorial and reviews actually, genetics and genetic improvement program has a lot of benefits with the societal changes, nutrition security, food security, employment, and then earning, foreign earnings. So these two reviews can tell you exactly what is happening. If you look at the aquaculture and economic growth, this is like you call it a blue economy, blue product, blue revolution, everything. But if you look at this value chain, aquaculture can give you many options. You have a seed, you have a feedstuffs, you have a feed mill, you have farmer, transporter, retailer, consumer, and so all can be done by like you can have a hatchery, aqua feeds, traders, grow fields, transport of fish fry. This is some of the people can get into this, and access to affordable fish. It's not like other high demanded like consumer product, right? Anybody can offer whether it's a fresh practice or not. As you know, like the 2050, you have a people less than of 9 billion people, 10 billion people. And what is the gap? You have a 56% food gap, you have a 593 million hectare land gap, and you have 11 gigaton of greenhouse gas mitigation gap, right? So if you look into the consumer, like how the people across the globe, this all indicates a different percentage of consumption of fish, and the wheat comes in around 10 to 20% animal protein consumption. But if you look at some of the countries, they're more impact actually. And if you look at the present status of this Indian aquaculture, we are somewhere around 12.59, but as of today, we are around 13.89 million metric ton production. But the point what I'm telling you, we are still in 11 kg where the demand is 20 kilogram per capita consumption. Okay. But one positive point is the growth rate. The growth rate actually, if you look in the inland, it is around 14.5%, 0.5%. Marine is very poor because, almost you might be knowing the stock decrease. How do you do actually the research as per the aquaculture fisheries is concerned? We look into biology, we look into omics science, you might know transcriptomics, metabolomics, like the proteomics, genomics. Then when you combine biology and omics, there is the possibility of getting innovation. So once you get innovation, you need to go for technology. Or you need to patent your, protect your patent right? Then biosecurity. Suppose if you are doing some sorts of like, new species, there is a high possibility of exotic pathogens to come into India. Communication, what do you do it in Kashmir, do it in Agartala, do it in Chennai, and you need to communicate to the people, outreach. The technology what you develop, it need to go to the mass. So this is again, I don't want to talk much about aquaculture, but the question is you can see the nutritional value of different meat products. Look into salmon oil, ham, carp, tilapia, chicken and beef. Okay, if you look into this essential fatty acid, what you are having maximum in the fishes. So this is salmon, the carp or tilapia. And you have less in the chicken and the beef. The point is why genetics is important. Deals with molecular structure and function of gene behavior, pedigree information, you can get information of the whole family, genetic factors, genetic disorder, and genetically modified organism, and genetically improved crops and animals, right? Again, why fisheries in it? Genetics is important. Since we have been talking about like, this sustainability. So fisheries and aquaculture can be ecological, environmentally sound, profitably, and socially community acceptable. So this is what we call sustainable aquaculture compared to other industries. Yes, I do agree because sustainability in case of brackish water aquaculture was in a question mark. When the question mark it was in 94-97, the Supreme Court gave a verdict to go for demolition of aquaculture farm because aquaculture was not sustainable. But the industry never looked back. They have moved forward. Now they have a sustainable technology for any farming, whether it's tilapia, pangasius, or vanamai, or monodon, or Indian weather crops. Okay. Genetics, why important fishery genetics? Increase productivity of fish and improve their productivity quality, future food, high protein, better amino acid profile, sustainable aquaculture. And you will be believe actually there are a lot of companies in Spain, Norway, USA, and the companies are coming to invest in the selected breeding. But as of now in Asian country, we don't have much investment from the company side. They're resource efficient and economical profitable as far as the genetics is concerned. So genetic mutation what it leads to? Uncontrolled capture fisheries, I told you the growth rate in marine fish is very low. Intensification of aquaculture, habitat destruction, infectious disease, what has happened in Indonesia during 2000-2002. And two key features that enhance the potential fish genetic improvement, that is fish remain in the early stages of the domestication process, which is linked to higher within the species genetic diversity. It's not like other animals. Secondly, they are highly fecund with a typical external fertilization, lower generation introduce. Take the example of geographies. You can get egg every day. Take the example of a tilapia. Take the example of kamakna, right? So you have a generation gap is very less actually. So it is easy to get the your required product within a certain time in the consumption. Okay, genetics plays an important role. Selective breeding, then you have a different trait. I'll come to that. The time lag between domestication and selective breeding considers certain in aquaculture species, which I told actually in the earlier classes. So how do you plan our genetics program, right? So we need to have a mating design to decide and a tagging method you need to tag to get the predictive family information. Then selection method, you are going for dialing cross and how you are going for hybridization. Okay. But what you need to have a base population growth genetic variation. What we did for Katla, what we did for Doho, what we did for the Scampi actually. You need to collect the animals from the different geographical locations. Those are showing a potential genetic variation. That will give you the best growth. Then protocol for recording traits, selection of booster, estimation of genetic parameter, and dissemination input stock on the brain. This is more important actually. If you look into the dissemination plant like John Kiroho, we have a multiplier unit, we have a breeding nucleus. So those who take care of the dissemination. So basic model of genetics is where P is equal to G plus E. phenotypic variance, genetic variance, environmental variance. This is more important when you go for selective breeding program actually. Because the genetic and input variety has to behave properly in every location as far the growth is concerned. But if the environmental factors are included, then there might be changes. Right? So what happens when G becomes genetic variance? What is A plus D plus I? Additive genetic variance, dominance variation, and epistatic interaction, interallelic interaction. Right? So these are like the basic model of genetics when you start like selection program. So what are the quantitative traits? Qualitative and quantitative. The qualitative traits are these are controlled by one or two genes, or quantitative is controlled by many genes. Qualitative phenotypes are the phenotype that are described as color, scale, pattern. Quantitative are those that are measured as a length and weight. They show discrete variation, but this continuous variation in the quantity. They are less affected by environment, but they are highly influenced by environment. Right? You may not have a problem with muscle flesh, but you may have a problem with the parasites or any disease which is prone to the flesh. So earlier what used to do is to do hybridization, genome manipulation, selective breeding, molecular level assistance. Right? What we are like recently we are looking at marker assisted selection, genomic selection actually. So if you look at the selective breeding in Norway, the theory or the history of selective breeding around 1970, like this J.Drum and Professor Skebold, this is a permission to capture genetics strain of Atlantic salmons from all different 40 different growth along coast in order to establish selective breeding program of Atlantic salmon. So 1970s families were established from 12 members, and this Professor J.Drum, who died in the 2020 July. So he is the father of selective breeding. And we are fortunate to have him at the ICAR-Central Bhubaneswar. Okay. So what we did actually, selective breeding we started in 1992. Labia rohita was a species chosen, and we took only growth as a trait. But after that, we have added disease resistance as a trait. And it's called as John B because it was released during the commemoration of the 50th anniversary of Indian independence. Okay. So, you can see it like selection differential, selection S is population mean and mean of selected parent respond to selection. You can see this is like, your best population and during that best population, you see the growth pattern. But if you look into when you go for selected animals, mean of selected animals, this in the generation one, you have a higher growth. This parabola has moved to this side, you can see the selection difference and the selection response. The growth response, you can see the changes, right? So genetic research strategy, what you are doing, genomic strategy, I told chromosome manipulation, conventional breeding, combination strategy, then gene transfer, GMO, crossbreeding, and gene editing. So this is CRISPR-Cas9 in the recent technology has been used, has been successful in case of the plant science. So now slowly it is getting into the fish research actually. So what happens? How you go for a genome and chromosome manipulation of fish? One is gynogenesis, androgenesis, polyploidy. So this is more of like developing a female-related population, and this is more of male-related polyploid, which is given a different number of chromosome manipulation trying to be tetraploid. So this is what like an induction of meiotic mitotic dialysis in the pioneer model. You can see that how it happens. Irradiated sperm nucleus, sub-treatment, gynogenesis, first place, sub-treatment. So diploid mitotic, you can get a gynogenetic individual, right? And the androgenetic same year pioneer model, irradiated egg with inactivated nuclear fertilization, egg nuclear degenerate, polar body extrovert, first clearance sub-treatment, and you get a diploid androgen individuals. So utility of gynogenesis, androgen in aquaculture. Gynogenesis and androgenesis provide a shorter route and an effective method to build up homozygous inbreed lines in a faster way. By top crossing these inbreed lines with heterozygous stock, progeny with higher growth rate, 30 to 40% over the normal one can be expected. So you can see the difference. Why should you go for androgenesis, gynogenesis? Production of monosex population is also possible through gynogenesis, which I told about, male specific to male specific. Male tilapia grows faster than female, so monosex population of male through androgenesis can be utilized to increase the productivity. Okay. So whichever gender you need, you can go for that. Polyploidy, Again, manipulation of chromosome number to give polyploid individuals is common in aquaculture. The retention of the second polar body during the second mating division in the oocytes results in triploid individual, which have two chromosomes set from the mother and one from the father. So, what is the utility? Sterility in commercial crystals, advantage, abundance of reproduction in the farm species. Triploid individuals are expected to be at least grammatically sterile because of failure of homologous chromosome to synapse correctly at the first meiosis. Triploid common calf have shown more than 60% growth rate, right? This is the result. So what happens? How do you do possible matings? And if you go for this type of choice, you can see what is the inbreeding purpose. You know, inbreeding means you will have a loss of genetic potential. If you go for parent offspring, you have a 25%. If you have a brother, sister, full shift, 25%. Half brother, half sister, half shifts, 12.5%. Grandparents, grandchild, 12.5%. Aunt and nephew, 12.5%. But if you do like this, if you do totally unrelated mating, you have a 0% inbreeding. That's what we do in the selection program. What we did for jointly Rohu, we have the Ganga, Jamuna, local means Sita, Brahmaputra, Satluj. So what we did, female parent from Ganga we took, from Yamuna we took, local we took, like the female parent, Brahmaputra we took, again local, against Brahmaputra we took, Brahmaputra, Satluj. So what has happened? You go for a diallele cross, and you can see the size. This is farm size: Brahmaputra, Satluj, Yamuna, Gomati, Ganga. And out of this, we could get actually the best individual. So what happened? Our best population 93, 94. You will have a best population from year wise, right? We got it from local 93, Ganga 93, and again Ganga we got 94, Gomati 94, and rest of the riverine system the 94. What we do actually, we collect the this good stuff, and stripping of male female, then collection of milk and eggs. We do artificial fertilization, and these germs are for individual family. Then stock family the nursery for tagging individual and study the growth rate. So this is the normal workflow for Rohu and Katla in ICAR-Central. So this is again general workflow. You go for pit tagging with anesthesia MS222. You go for length weight. Then you study the growth pattern in the year in the particular culture period. So with this all experimentation, we could see you can see the normal row and jointing of the growth difference, right? So as on today, after 12 generation, we have 18% genetic gain from the jointing. Okay, so this is not required. This is what I was told in the 18% genetic gain at the end of 12 generation. Okay, so what the benefit we have got is the restriction of genetic and period stock, reduction in the rearing period at the 17%. Increase yield, you have a 24% increase, reduction in growing period 53 days, relative reduction in cost of a local stock. Around 20% you have less expenditure compared to local stock. So this is what if you summarize this whole selection program actually, you have a founder population 6 like and the one from ICAR-Central, you had a diallele cross mating design, full C families, base population G1 we get 70% genetic gain, but at the end of G12 generation 12 we have 18%. This is cumulative genetic gain. And Katla in the same process we are doing now at the end of second generation we have a 15% genetic gain. This was done under project called DBT Center of Excellence, which I was leading the project. Scampi, Dr. Binduar Pillai is doing it. So we get a 77% genetic gain at the end of the 12 years. And we did a comparison between the Katlas and the normal row, right? So what has happened, you can see actually polyculture with joint row and improved Katla, and the polyculture normal row and normal Katla, right? You can see everywhere you have a higher benefit. Growth, average growth, survival is there is a not much difference, but you have a production, you have a difference. Cost of production, the total cost of production, but the point is, you have a benefit cost ratio of 1.7 compared to the normal growth and the normal fertilizer, which is 1.4%. And if you look into comparison between Rohu and Katla, this is the best population, rate of selection, method of selection, start of selection, generation selection, heritage estimate, genetic gain for generation. Even I told earlier, but you can see actually We started in 1992 Rohu, 2021, 2011 Katla, and GI's campus at 2007. And here it is gone, 12th generation, 3rd generation, 4th generation. If you look at genetic gain, 18% in case of Rohu, 15% Katla, 7% in case of Scampi. This is just summarization of what has happened genetic program. And this is again, we have developed a natural selection process, signing bar, which is a different coloration. And this is different in the normal strategy actually. What has happened? We breed the seed here, give it to the multiplier. Multiplier gives spawn back sugar to the farming practitioner, right? So they go for culturing. Coming back to tilapia, you all might be knowing tilapia has been ruling the world because being a wild fish, people are preferring tilapia. And as of today, at the end of G6, this generation 6, so they have their combined allele program, and tilapia has been having around hardly 8 to 15% genetic gain after 6 generations. It's almost all equal like what we have got in the Katla in the second generation actually. So as I told, this food fish in the 21st century, tilapia is really across globe. And this genetic program I told already, Rohu and the Scampi. I just want to tell you, we have taken additional this one trait as a survival against the Aeromonas hydrophila. Already body weight I talked about. Scampi also harvest weight, carcass length, and standard length. These are the parameters. But the point is, we have a trait like growth and survival. And if you look in Magur, CIP has taken over. They are exclusively working on the growth. And the tiger prawn, again, it was from CIP and the SIPA could not be successful. And if you look into global component, actually, look into this salmon. They have almost around 6 to 7 trade, right? So it's become difficult for us to compete with them unless we go for a much more trade, actually. And their rainbow trout, you can see they have 1, 2, 3, 4 trade. Common carp also, you are having 2 trades. Again, tilapia, just look into growth, and the channel catfish also growth. And if you see the relative increase around growth is around 25% in case of harvest weight, and in case of channel catfish, growth is almost around 28% increase actually. So coming back to genomics actually, look into this particular image. You might be knowing salmon. Salmon having a issue of growth during the winter season. So what they did actually, they did a gene editing or genome manipulation program. You can see the image actually. This green bar talks about aquatic advanced salmon. I'll tell you how they have come out. And this is standard salmon. The point what is aquatic advanced salmons get a weight of 6,000 gram at the time of around 700 days. Whereas the normal salmon grows up to maximum 5,000 gram at the end of 50 days, right? So what they did actually, they brought Chinook salmon growth hormone, and they took ill pouch promoter. They added it into the Atlantic salmon. So because of that, they could get a higher growth rate. Okay. Again, coming back to first commercial project, genome editing tilapia. And this tilapia, genome edited tilapia has come. So what has happened? The benefit 70% of significant improvement in the fillet yield, 16% growth rate improvement, 14% feed conversion rate improvement, shortening of the feeding time. The fish no need to spend much of time for the feeding. So it will be possible to reduce the disease risk and decrease the input cost. So this will facilitate a sustainable solution for world's protein gap. And recently tilapia had a big issue of tilapia lake virus. So the applying genomic science, they could get the chromosome Oreochromis niloticus 22 have that particular issues for the promoting this particular disease. So now the possibility of breeding tilapia resistant to tilapia lake virus is one step closer thanks to genomic research breakthrough. Right. So once you know the loci where the chromosome is the gene is responsible, so they could do it through a gene editing technology. CRISPR-Cas editing, what I was talking about, this is what our project, I am also involved in this. We are working on CRISPR-Cas edited to test and control genes implicated in influence aeromonas disease resistance in carp and salmon. So Norway team is working with salmon, we are working with rohu, right? And again, we are working with genetically modified for the translucent freshwater fish. We want to go for customization of the color. And some of the initial, we have some success in case of rohu and a zebra fish. Then we did this is more about two-step protocol for rose format stem cells investment was developed. We wanted to see whether artificial sperm cells can be formed or not. We could get it in case of agar also, but the fertilization rate has been very poor. So if you look at the genomics in the fish health management, in common crop resistance, so Acanthomices invadens 5,288 genes were found to be different and expressed, of which 73 were involved in the 21 immunopathways. Right? So this is application of genomics. You can understand the pathology, pathways, and a innovation strategy also you can understand. But accordingly, you can plan your remedial measure also. And again, this available the complete pathogen genome sequence with the contemporary development of bioinformatics tools has led to a new paradigm of vaccine development. You might be knowing Corona has given a big scope for all of us to talk about a new product and new vaccine, right? So in aquaculture also, we have a pan-genomic reverse vaccinology technology, host pathogen introduction, and engineering of the potential antigens actually. So this is the process, how the vaccine is developed. So no need to go into details. Somebody else will be talking. The point what I meant to say actually, the vaccines is possible in case of aquaculture also. And again, if you might be knowing about the microRNA, which is short molecule involved in the most transcriptional factor actually. So we are working in a particular species called Chana morulus. Chana morulus, what we could see actually, we can see like the different microRNAs are playing a different role in case of the reproduction in case Chana. So we could see microRNA 34A, 133B, microRNA 200, 21, 22, and 205P are eventually reproduction. So what we did actually, if you look in the microRNA, as I told, the microRNA translation, how it is happening actually, transcription mRNA gene, and this you have a microRNA database. If you look into this MIR, denotes the name of the precursor sequence, where MIR is denotes the name of the mature sequence. And the first three letters represent the species name, that is DRE, this Denio radio microRNA 34. So what we did actually, we could see the significant difference in the expression of the mRNA in the males and females across the pre-spawning, spawning, and post-spawning season. You can see this microRNA 34, 32025P, 21, and then you can see that this is in the male gonad, this is in the female gonad, and this is in the male gonad, again this is in the female gonad, right? So you can see the difference of the expression pattern actually, pre-spawning, spawning, and post-spawning. When you talked about the pre-spawning phase, means almost the phase is ready to have the matured egg. Spawning means almost it is in the ready to stage of spawn. And the post-spawning effect, you can see that how post-spawning is having a impact of this particular microRNA. And we are still, again, we look in the genomics. I talked about the transcriptomics, I talked about microRNA. Now we are more into metagenome analysis. The point is what we want to understand actually how the microbes are playing a role in the brain fish reproduction. So there are a few interesting studies which a particular lactobacillus can influence the gender difference in case of zebra fish, right? So we are more into that metagenome analysis. We are working on again on the Chona and zebrafish and anabas. You could see the Cetobacterium involved in vitamin B2 synthesis was most abundant genus, followed by Endoheterobacter and bacteria on placenta. Right? So this is what metagenomics talked about, and the Clostridium as a probiotics for improving growth human response was the most abundant genus, followed by Cetobacterium and Lactobacillus in T2, where bacteria unclassified were the most abundant genera in Q1, but followed by Enterobacteria. So what happens actually, you have it's a blood sample, a skin sample. You can know the abundance. And once you know the abundance, you can go for functional annotation, how this particular group of microbes are playing role in case of reproduction. So this is, what we did actually, we did an environmental manipulation. So when you do environmental manipulation, you can see that, this pathogenic microbes shows less problem, whereas you have a beneficial microbes are there. You go for habitat manipulation. So what we did, we gave a natural environment to be mimicked in that particular period. And if you look into the overall this research of this species and the genomics and the genetics and biotechnology. So I have taken a rohu as a model fish, what we have been working actually. So we are looking into genetics, inclusion of more traits. We are looking for high fecundity. We are looking for what is a good quality fortified fish. We are looking for a short reproduction process, right? And phenotyping and sterile reproduction. These are the new traits we are looking for genetics for. If you look at the genomics, so we want to have a QTL identification for Aeromonas hydrophila, and we have to use the SNP panels what we got from whole genome sequence in the selection program. Which SNP is much more beneficial to have a better growth. Then we are going for genome-wide association study for genomic selection. Look into that, and sometimes we can go back to the old population also how to, during selection process, many things have changed. Epigenetics, tissue specific microRNA, how the temperature is playing role, then general genetic and environment interaction. Then in the metagenomics, I talked about identification of microbes linked to health, nutrition, reproduction, probiotic development, antimicrobial resistance. Right? Transgenics and genome editing, development of autotransgenic rohu, CRISPR-Cas9 gene edited rohu for European disease management. Again, transcriptomics, tissue-specific gene discovery, differential expression of genes during development of biology, network pathway detection, novel gene identification. This we have done quite a good amount of work by which transcriptomics plays a major role, and we can know how a fish is breeding during, breeding season or not breeding during the non-breeding season. So what is need to be done actually? We have to strengthen our research into fish health management, doubling fish production through diversification of species, new age entrepreneurship, global food security, genomic approach, fish for Cas9, optimization of water use and increase water productivity, science communication, what I told you in the morning session, we need to communicate very frequently. Then integration of fish in country's nutrition program, that's what we need to skill. Sorry. So if you summarize everything, we have a traditional breeding. You have a phenotype, dissemination, breeding nucleus, satellite center, hatchery. Sorry. If you have marker asset breeding program, you have a genetic variation, polymorphic parents. So from there, mapping population linkage and QTL map, marker maps, physical maps, then computational map, genomic resources. Then you have a QTL tagging, and finally you get a candidate gene, superior QTL, and you get a designed genotyping. So this is what done through the marker asset program. And we will come at the transgenic candidate genes, promoters, like what aquatic salmon have done. You have a gene delivery mechanism, integration study. And once it is aligned, then germline transmission, you might be knowing how the surrogacy is being done. Then finally you get a variety of breed. So this is all we have made for the farmer or farming population. Okay. So thank you very much. This is what the overview of my presentation, and this is what I will talk about grow fish, grow with fish. This is SIPA's overview. Those who are already visited, you can come once again. Those are not come, please visit to us. Thank you very much, Mr. Hamid Khanna.
Thank you, sir. Thanks a lot. Now, if there are any questions from the questions or interactions from the participants, they can turn on their microphone or send us their queries in interactions via chat also. So please, participants are requested, if they have any question or query or even an interaction they want to do, they can turn on their microphones and turn on their cameras and pose the query. Please start.
I hope there is no more queries. If any queries comes, I can answer them by mail.
There's a query in chat. If you
Yeah.
can click on the chat, yeah, sir. Click on the chat, yeah, perfect. Click on it. There's a query from
If
S.
any case which are developed through transgenic approach, if it is yes, what kind of trait will be developed, please? See, this is mostly transgenic. They go for higher growth rate or disease resistance. Okay, so what Aquaculture salmon, which was developed for transgenic, it is more about growth because smolting problem with the salmon. So that is one thing. And yeah, anything else? Trait will be growth and disease resistance. Thank
If
you,
there is
Mr.
any
Sagar.
other question, please ask.
Sir, good morning, sir. Sir, any
Yeah.
ethical issue regarding the development of transgenics?
See, government of India has yet to give the permission, but European Union is not favoring. But UK, USA, I think some countries they started moving with the transgenic. And but government of India has given a ban on any transgenic, which is a plant science and animal science. But research level you can do. Ethical issue will be there because it's all about how do you develop your science and communicate to the people. If you see that your transgenic products are not getting into the exposed to the outside, there is no ethical issue. But if it is goes again breed with your local population, then a lot of issues will be there. As usual, transgenic having a social taboo, right?
Yes, sir. But government of India, they are not at all allowed for the food
Yeah, yeah, that
food-based
was good.
GMOs.
Yeah, but the point is, research cannot be stopped actually.
Yes,
You
sir.
cannot release your product, but you can still continue to work on that.
Once if you develop the technology, so how
Mhm.
long it will reach to the commercialization, sir?
Which technology?
GMO fish.
See, this is all dependent on, it's all the life cycle actually. This is, suppose you take the example of Katla Rohu, it gets a maturity in the 2 year plus, 3 year plus, right? So once you develop a transgenic product, it has to complete a life cycle, right? If you are going for sterility, it's okay. So want to have a next further generation, then it takes minimum 3 to 5 years and standardization, then ethical clearance. So, maturing a technology depends upon how fast you are in the research and how authentic in your research. Because when you go for transgenic product, there are a lot of questions will be there. You will be in a position to answer all those things, right?
So any product will develop, sir. Already any commercialized any product will be developed in India.
No, no, no.
GMO fish.
See, if you look at the plant size, you have a lot of GMOs. Like you have a okra, you have a brinjal.
Yes,
But
sir.
in animal science, fishery science, GMO products are not there.
Yes, sir. Okay, sir. Thank you, sir.
Thank you, thank you.
Any question or query? There's a thank you and very informative session. Thank you by Dr. Ajay Kulkarni. Anyone who would want to ask any other question or query or even say a thank you can turn on their microphone and go ahead. We have few minutes of our time. Dr. Ashwini Kumar is saying thank you in chat. Anybody else would want to say some words, share some experience? We have few minutes. If we have any queries, questions, inputs, suggestions, feedback, all are welcome. Sir, I think there are no further questions, and I think
Okay,
like we can consider the session to be concluded.
okay. Thank you
On
very
behalf of the participants and on behalf of the organizing committee, I'd want to extend our heartfelt gratitude to you, sir, for being with us, for sharing this lovely presentation with us and the concept and the interaction at the end. We're thankful to you, sir. It was nice, so nice of you. And we look forward to basically working with you in future these
much. Okay,
sessions. Thank you.
Thank you.
Thank you, everyone.